So we're going to talk about organization in terms of information, how that's expressed. So we're multicellular. That means that we're not a single cell. We're many, many, many billions of cells. And that's not even counting all the bacteria that are in us and other kinds of things. So we're made up of cells. Trillions? I guess so, yeah, okay. Now, in the middle of each of those cells, since we're eukaryotic, there's a nucleus. And in the nucleus, what's in the nucleus? Anybody? DNA. So, that DNA is actually on the chromosomes. And for each chromosome, there's actually two chromosomes. There's a pair. And these are identical in terms of the genes that are on that chromosome. They're identical not only in that it is exactly the same place. Okay? And we can actually look and see. So, in fact, here. Okay. That's a locus. This is the locus. This is the locus. Now, we've got 22 identical pairs, and then we have a pair of 6 chromosomes that are either an X or a Y. If you're female, you're XX. If you're male, you're XY. So if you're going to order this from smallest to largest, would you start with a chromosome pair, a genome, a cell, or an organism? Smallest. Anybody? Yeah. And if you wanted to go down even smaller, you would go towards gene. Each of those bands is a genetic locus. Okay? So, these are genes. And each chromosome is one long DNA molecule. Now, it turns out we also have DNA with our mitochondria. Mitochondria has its own genome because the eukaryotic cell is actually a combination of a bacteria and a eukaryote with a nucleus. But for now, we'll just go on. Okay, so along the chromosome are genes. Now, not all of the chromosome is actually made up of genes. So, okay, so DNA is a double helix. And the genes are composed of these base pairs called nucleotides. There's 4 of them. There's A is adenine, T is thymine, C goes with G, that's cytosine and guanine. There are no other base pairs, there's just 4. And if you get 3 of those nucleotides together, like A, C, C, you end up with what's called an amino acid. And what do amino acids make? Proteins, exactly. Okay, so let's take a look at DNA. Our chromosomes are made up of DNA. The backbones are made up of sugars and phosphates. Okay, these things right here. So if we go to the, there we go. So I'm tracing 1/2 of the double helix, and these are made up of sugars and phosphates. There's a complementary strand made up of complementary nucleotides. That is the other part of the backbone of sugars and phosphates. The backbones are all the same. They don't vary it at all. The actual genetic information is the nucleotides made up of the 4 base pairs. Okay? So, if that's an A, what's its complement? A T or a C or a G? T. Okay. Okay. Okay, so that's how it's set up. Now, if you only had one of the strands out of the 2, you could make it a copy of that very easily, couldn't you? You take the strand, let's say, on the right with the A and the complement. You create another strand, and the base pairs, you would put a T, a G, whatever the next one was. Let's say it was another C, you put another G. You could duplicate it very simply just by going according to the base pair strand of what it was. And those strands, those base pairs, they're nitrogen. They're called nucleotides, okay? And like I said, there's 4 of them. So if you were to split this molecule of this chromosome of DNA, you could easily take one of the strands and make a perfect copy, and then have an identical chromosome. And in fact, since our body is always making new cells as the cells wear out, things like that, you have cell replication. And that's how cells replicate themselves exactly. Okay, so this is what it looks like. Let's see, here is the phosphate sugar backbones. And then each of those is a complementary strand that is tied together in the center. And the nucleotides are the things that actually carry the genetic information that is transcribed and translated into a protein, which is made up of amino acids. Now, each amino acid is coded by 3 nucleotides. So in this case, ACG corresponds to a very specific 1 of 20 amino acids. And then when you string all these amino acids together, then you have a protein. Okay? So these 3 base pairs code for 1 amino acid. Now, all of our chromosomes, the total base pairs is around 3 billion for human beings. There are some organisms that have fewer chromosomes or more chromosomes. Like, I think corn has, let's see, what does it have? How many chromosomes? I forget, like 40 or 50. So corn actually has more base pairs than we do. That means corn is more advanced than we are. Not really. So I just want you to make sure that you understand that just because I have more than you doesn't mean that I'm better than you, even though we all know that I am. Okay. Furthermore, there's one other thing. Okay, so we have all these different base pairs. Now, it turns out, though, that the coding regions, these triplet areas that code for, made up of nucleotides that code for amino acids, only make up about 2% of our DNA. For a very long time, it was thought that the rest of the DNA is junk DNA. And some of it, I guess you could say, is junk DNA. You can look and you can see that there are genes that are broken. They're called pseudogenes. And they might have, let's say, a stop codon or some problem in there. They're still in there, but they're not actually coding for anything. And there's also these genetic elements called transposons. And what they do is they copy themselves from one part of the DNA to another. We say that they're kind of parasitic elements because if you copy a transposon from one place to another, if it lands in the middle of a gene, you might make the gene non-functional. So why is it there? Well, it turns out there's ways to stop that from happening. Also, transposons, it's becoming apparent, are actually important in a lot of times gene regulation. In other words, turning genes on or off. So even though only about 2% actually codes for proteins, there's a lot of the DNA that actually is regulating the coding of that DNA. In other words, and when you're making proteins, you make a certain amount. Well, you can dial it up or dial it down. Or you can even turn it off, like we saw with the lactase gene. When mammals reach a certain age, in other words, they're being weaned by their mother off of the mother's milk, the individual doesn't need lactase anymore to actually being able to use the lactose molecule, and so it's shut off, except in humans who actually are lactose tolerant. Okay. There's a little bit of a DNA, let's see, Watson and Crick. This is back in the late 40s, early 50s. At the time, people started to get the idea that perhaps DNA was the molecule of inheritance, but they didn't really know that. And so they were trying to figure out the structure of this molecule. And Watson and Crick, one was an American, Watson and Crick was English, and they were both full of themselves, and they decided they were going to figure it out. Now, there were other people that were trying to figure it out too, so there was a race to figure out the structure of DNA. And as you can see, that's a model, and they basically, if anybody's ever taken chemistry, a lot of times you make models of molecules. So they made this model. There was a woman named Franklin who was using something called X-ray crystallography, which could tell what elements were part of a molecule and how those elements diffracted so they can get an idea of what it looked like. When she got that information, they saw it and they suddenly were able to figure out that in fact DNA was a double helix, which is what we've been looking at, these double strands that curve around each other. And they were able to figure out the structure of DNA. Now, some people thought at the time that perhaps DNA was a triple helix. The thing that was confusing was that all of these instructions were on the inside of the helix, not on the outside, so that was confusing. They didn't realize then that the strands separate. So they figured it out, and they basically got a Nobel Prize for that in 1953. Now, Crick went on. Once you had the structure, you still had to figure out how it actually coded for various proteins. And Crick was the one who then subsequently went on and discovered that, the coding system. In other words, these different nucleotides that go together, A and C, T and A, and then realizing that you need 3 of those nucleotides code for an amino acid, and then those amino acids are strung together. So he figured that out as well. Watson's still around. He's in his 90s now. So here we are. We saw that little video. So we have a DNA molecule, a double helix, right? What it does is it unwinds. When you see ASE, what does that mean? What kind of protein is that? Anybody? Anytime something with ASE ends with ASE, it means it's a certain kind of protein. It's an enzyme. Okay? That means it has a functional domain that actually makes things happen. Okay? There are some proteins that are structural, that are a certain molecule like hemoglobin that are made out of, but those are coded by DNA, and that has to be translated and transcribed. So DNA polymerase is the enzyme, and then as you have these single strands, separate, you can now make a complementary strand, can't you? So for each one of these, the complement would be right here for this new, so each strand will form a new strand, and this, so you'll end up having 2 complete double-stranded helixes if you want to copy it. And you can easily make the copy, if this was, let's say, an A, we know that this over here would be a T. Right there, right? Oops, that's nice. Oh, come on. Oh, well. Okay, so. And now we form 2 identical double helix strands. And when the cell replicates, each cell gets a full DNA of all the different chromosomes. Okay? Sometimes there are some errors in there. Those are called mutations. Okay, and that's what we've already. Okay, we're going to get back to this in much more detail, but for now, this DNA is basically a way to, it's a memory system for information on how to make various proteins. Now remember, in terms of species are species that are more closely related. Are they going to have more similar DNA in terms of alleles or less similar? Huh? Yes, exactly. Well, the same thing happens in terms of us and our parents, or us and our relatives. If we're more closely related to someone, we are going to share more of our DNA in terms of the identical alleles because we've basically inherited those from previous ancestors. So, if I have, let's say, a sister who, I told her, of course, that she was adopted, so that she was messed up for the rest of her life. If that was the case, then we would not share any DNA except by chance. But if she is actually my sister, then we should share 50%. In other words, we should have the same alleles along that DNA because we inherited those same exact copies from our parents. So I'm 50% related to my sister. If she was adopted, I wouldn't be related at all, or very little if you have to go back, 1,000 generations perhaps. I'm related to my parents by 50%. I'm related to my cousins, it turns out, by 12.5%, 1/8. You don't have to know that right now, but so you can see that the more closely related you are to each other, the more closely your DNA should match. Now, that lets us actually figure out things. Anybody watch CSI? Okay, right? Okay. It's all a bunch of BS, actually. But anyway, there's something called forensic science. It's actually not science. They like to put a science there, but it doesn't have much to do with science at all. But anyway, you can take DNA analysis. If you collect DNA from someone, collect their cells, you can figure out, first of all, who they are. if there's a copy of their DNA in some database. So let's say they're a criminal. And you've got this woman who's been raped. There's semen that's collected. And they take the semen, which is full of sperm, and they can figure out the DNA sequence of who the father is, and they might be able to compare it in a big database to all these other people that have their DNA stored, because let's say they've been convicted of something. Now, if the person who did this has never been caught before and had their DNA, they won't be able to find a match. But let's say they've got a suspect. And so what they do is they've got the suspect, they take a swab in the mouth, collect some cells, sequence the DNA, then they take a sample that was collected during the rape kit, and they do the sequence for that. If they match, they can say with a great deal of certainty that, oh, this is a suspect that actually raped this woman. Well, he had sex with this woman. He may say it was consensual, but we know he's lying, right? He's a scumbag. Okay, now, so we can use that to solve crimes like that if we can get DNA. We can identify human remains because if you remember, we're more closely related to our relatives. So let's say we have a mass grave in Iraq. There's all these bones and remains of humans. They want to find out who is in this grave, who's been murdered, and they might go and start collecting DNA from people who are missing relatives. People who are more closely related should share more of their DNA, and so they can compare the DNA from various bones and figure out who's more related, like perhaps someone is the son of a mother, so at least half their DNA should be identical by descent. Furthermore, you could have missing persons, disasters like an airline crash or something like that. You might want to find out who the father is in terms of an infant. The mother says it's him, and he says it's not me. Well, they compare his DNA to that of the baby's and see whether, in fact, he is the father. So you can use DNA analysis, and I would say it's one of the first things that really gives you evidence that you can use to differentiate between people. Turns out that fingerprints aren't nearly as good as they were thought. You can't really match bullets to a gun. You keep hearing that, but you can't. Every time they take experts and say, who did this fingerprint sample come from, or who is this bullet, was this fired by this gun that this person had? When they start testing that, they find out that there's usually 10 or 20% errors. Well, that's not good enough, right? Okay. But DNA is pretty good. Okay, so turns out all of you right now are leaving samples of DNA on the chair, on your computer. You go out here on the door, all that kind of stuff. Because we leave cells all over the place. You can get DNA, let's say you have an individual who's been raped, and she fought the individual, and there's skin samples underneath her nails, saliva from an envelope, skin on dirty clothes, saliva on a cigarette butt. You can collect DNA on all sorts of different places. Now, in the past, you needed a bunch of cells to do this, but now we can just take a very, very, very small amount, in fact, one cell, and copy that DNA and make many multiple copies of it. And that's called polymerase chain reaction. Now, ACE, again, it's an enzyme. And PCR, as it's called, allows you to make copies of a target sequence. Let's say this sequence right here, between here, here and there. Okay? And at this university, this is a very common procedure. Lots of people do it all the time. We do it all the time, not just for forensics, but for all sorts of different things about figuring out function and things like that, where you need many copies of DNA. Basically what you do is you take this double helix and you heat it, and these separate strands that used to be next to each other, tied together with these base pairs, they come apart. You can then take these bases, primers, that are on the either end of the target sequence. These don't change. A primer is always the same. They're not variable. But in between the actual sequence that you want, let's say, in between here, there's lots of variability. And so we can make copies. So we use these primers, we have a copy of that whole strand. Then we use PCR to take, in this case, one strand, we separate it with the heating, we make a complementary strand, and then these two make two more, and two more, and two more. And so, in no time, we can have millions of copies of that DNA. Exactly the same copies. There's no difference. The alleles are the same. So now it's a lot easier to work with. We've got millions of copies instead of one. Now, as I mentioned, only about 2% of our DNA actually is used to transcribe and translate the information on our DNA into proteins. There's all these other regions that we used to call junk that are oftentimes highly variable. In other words, they might be, let's say, a pseudogene, and there's a part where there's always the same, let's say, 3 or 4 letters, TACG, TACG. And they vary between people. Now, there's more in common with people who are more related to each other compared to other people. So there's all these different variable regions. And what we can do is we can find out and sequence the repeats. So in this case, now remember I said for DNA to be transcribed, you need 3 base pairs, but it doesn't matter. Here is this variable region, and this individual has 8 repeats of TAGC. Okay? Someone else might only have 4 of these repeats. Someone else might have 1. Someone else might have 16. Okay? Now, if this repeat is from someone who's related to the person, they inherited the same copy, that person who's related will have the same number of repeats. So we could say, oh, this is probably a relative, perhaps. The thing is, if you just had this one region that you were looking at, and one person had 8, one person had 4, well, there might be a lot of people in the world who had 4 repeats there, or 8 repeats, or 6 repeats, or whatever. And you wouldn't really be able to differentiate. You'd only be able to differentiate between groups of people who had 4 repeats and those who had 5. But if you take a bunch of these variable regions, let's say 20, the chances that they'd all have the same number of repeated nucleotides in each of those places becomes vanishingly small. So there's 8 here. This individual at another region might have 4, not necessarily of TAGC, could be something else. And another region might have 10. Well, if you take 20 of those, the chances of one person having exactly the same number of repeats at all of those different 20 regions becomes very, very, very small. Now, these repeats are called short tandem repeats. Nowadays, we use something different. We use something called SNPs, S-N-P, short nucleotide polymorphisms. You don't have to know what that means, but each of those base pairs, you can actually now sequence to the point where you get those base pairs, and you can get millions of those, or each one is separate. And so there's variation in those SNPs. A million of them, there's no way you're going to get one person with the exact same number of SNPs. But anyway, this is an older one where some people still use short tandem repeats. So here we are. We amplify, in this case, this repeat, or we amplify the DNA of SNPs, whatever it is, with PCR, polymerase chain reaction. The PCR primers don't vary, but in between the actual repeats, they do vary by individuals. And after we've made a bunch of copies, we can take that DNA that we've made millions of copies, and we can actually separate them out. And we can separate them out on something called gel electrophoresis. Now, and gel electrophoresis separates these repeats based on the length by passing an electrical current through this gel. And this takes you through the whole thing. If you notice, in terms of these STRs, short tandem repeats, the larger fragments, the ones with, let's say, 8 repeats versus 4 repeats, the larger fragments move more slowly from negative to positive. Okay? So, and the smaller move more quickly, or more further down in a shorter period of time. So in this, we put all these DNA, all this DNA that's been replicated into the gels, we run the current through it, and we get these bands. And then we can say, this person is more closely related to this person. Okay. Remember, each person is diploid. That means they've got 2 bands. They've got one from mom and one from dad. Sometimes mom and dad had the same fragment length or the same allele, and so you might get a thing where they have the same size band. Other times there'll be like person number 2. We've got a band that's longer and heavier up the top and shorter down at the bottom. But everybody's going to have 2 bands. One from mom and one from pop, whether it's a double band or not. So person 6 is another double band, right? Otherwise, they've got separate. Okay. So you can see that on person number 2, there's a longer, heavier band up the top. It moves more slowly down the PCR versus the other one. Okay. So who's not the father? Okay, so here's the baby. Mom has these 2 bands. Which one does she share with her baby? This one. So, we know that the father contributed this one. We want to know who's not the father. So we go back. Here's the baby, his father. And we say, oh, 2 and 3 both have the same bands. Either of those could be the father. But we want to know who's not the father. We can say, well, number 1 does not have that band, and so it's obviously going to be that individual. Okay? Now you can say, well, why do they share the same band? Well, remember, this is only one variable region, one locus, when we're going to probably have 20 to compare, or even more than that. Okay? But this is how you can actually figure things out. In the past, what we did was we had the FBI used 13. And these are the 23 chromosomes, 22 that are identical, homologous. Right? And then the 6 chromosomes. And each one of these is one of those variable regions. Okay. So, in this case, who's the father? Well, first of all, let's get mom here. Mom has this allele and this allele. The baby has this allele. So the father has to have this allele. Right? Okay. Again, this is the father. This is the mother. Okay? We say, oh, it looks like number 3 is a father. Oops. Because it corresponds from this band to that band. Okay. Before, when they just used 13 of these STRs, the chances of an individual having the exact same fragment lengths over 13 of these different regions was 1 in a billion. If they use STRs now, it's like 20 or 30, so it's much lower probability. And when they use SNPs, it's even lower still. So there's no chance really, except for maybe 1 out of, a trillion, that 2 people would share the exact same DNA in terms of these regions. In fact, it wouldn't happen. Now, as I mentioned, a suspect in these investigations, or the remains of someone, that's basically a hypothesis. We say, I believe that Jimmy committed this crime. We've got the semen, we've got Jimmy, and we're going to test it. And the prediction is they should have similar DNA, or identical DNA. Turns out they don't. We've just falsified this hypothesis, just like any other hypothesis. Now, that's actually forensic science. That's science. That's what you're trying to do. You're trying to eliminate people. You're not trying to find a confirmation. You want to eliminate suspects. And that way, of course, you don't get the wrong person who goes to death row, and they kill them, and then they go, oops. Okay. So, what could this baby's STR pattern look like? Well, here's mom, and dad, and dad. And we could go 1, 2, 3, 4. Okay, we can say mom could be this, right? This band. Could also be mom. And then dad could be either this one or this one. Or it could be mom and dad and dad. So if you've got two people, there's two bands for each. You've got 4 possibilities. Here we are again, mom and dad. We can see that baby number 1, this could be the dad, and this could be the mom. This could also be the mom, or this could be the dad. And then all these other 4 possibilities. So if I gave you a mom and a dad, and I said, who's the baby? And I'd give you 4 possibilities, you should be able to figure it out fairly quickly. Now, remember we're diploid. There are only so many possibilities. You cannot have 3 bands. You can only have 2. You can't have 4 bands. You can only have 2. You can't have 1 band. You have to have 2. Okay. Is that it? Yeah, I guess that's it. So, I'm going to open up Top Hat for some homework. 4 questions, something like that. And I'll see you guys Thursday.
Do you have the tests here or no?
What?
Do you have the tests?
A test?
The exams, do you have them?
Yeah.
I didn't get them.
No, you don't get them back. But if you want to come in and look at it, you can.
When is your exam?
Right now, if you want.
I have a class on 15.
When are you free after that class?
Yeah, I have 1 hour.
Okay, then why don't you come in? It's in this building, second floor, 5218.
Thank you.
Sure. See ya.